My name is Elena Moore, and today I'm going to be talking about replication of DNA in prokaryotes. So the first thing we're going to have is RNA polymerase, which is a hollow enzyme, which means it's an active enzyme that needs help of a cofactor, which is the sigma factor, binds to the promoter region with the help of RNTPs, and the promoter region includes the -35 region, the -10 region, and the +1 region. And then towards the end of the DNA sequence, there will be a poly-A tail, which stops DNA if RNA doesn't stop before that, which you'll see what I'm talking about in a few. So initially, we have the helicase that breaks the bonds between the DNA bases. And then along with that, we have gyrase, which kind of goes through and makes sure that the DNA doesn't supercoil. And in the DNA, we also have what is called phosphodiester bonds, which are carbon bonds that bond each of these sugar molecules together to make up the DNA. And then, along with that, we have SSBPs, which stand for single-stranded binding proteins. So, between when the RNA is made and when the DNA is split, we have to have something that keeps the stranded DNA together, the single-stranded DNA together. So we have those. And then when Okazaki fragments aren't used, we have the DNA polymerase 3, which can just go from 5 to 3 and make the RNA. And then if it can't go 5 to 3 since it wants to go this way, but it needs to go that way, that is when we have the DNA polymerase to help with that, and the RNA primase to make those primers as well. And then the ligase will connect the Okazaki fragments together. And in the middle of this replication, so this replication group is the origin of replication. And then talking about the leading and lagging, the leading is the one that does not need help using Okazaki fragments. So as you can see, the RNA wants to go from 5 to 3, and that is possible. So this is the leading strand, and that is the lagging strand. And then, like I was saying before, instead of a poly-A tail to end this sequence, we have the termination hairpin. The template and the coding strands depend on which one you're trying to copy. So if you were trying to copy this, you would be that would be the template strand, and then that would be the coding strand, and vice versa. And then if you didn't have DNA polymerase functioning correctly, then you would not be able to make this top half RNA. And talking about ribosomes, if it was needed to make proteins, you could make the proteins from the RNA that was made.